Welcome to the Deep Dive. Today we're going deep into some bioengineering research.
Yeah, some really wild stuff.
So have you ever thought about, like, bringing a drawing to life?
Mm.
I mean, actually creating a creature based on a sketch?
Oh, wow. Yeah, I mean, it sounds like fantasy, but it might be closer than we think. The research we're looking at today proposes a whole system for doing just that.
Right, so the source material outlines this system that combines, like, super advanced tech with biology.
Hmm.
You know, it's kind of like grafting plants, combining different species to make something new, like a pear-apple hybrid.
Yeah, exactly. But instead of plants, we're talking about completely new life forms.
And this isn't just science fiction. This has massive implications for things like regenerative medicine and synthetic biology. I mean, this could totally revolutionize healthcare as we know it.
Definitely could. It really could.
So let's break this down. How does this system actually work? Like, how do we go from doodle to a real, living, breathing creature?
Okay, so picture this. You've got a drawing. Let's say it's a five-legged frog.
Okay, a five-legged frog.
So the system takes your drawing and analyzes it with AI.
Like facial recognition software.
Yeah, it's similar, but instead of a face, the AI is looking at biological structure. It makes a blueprint, instructions for how to build that organism.
So it's like the AI is translating the drawing into a language biology can understand.
Exactly. And then we have these things called circuit cells. They're like the building blocks of the system.
Circuit cells, what are those?
Think of them like microscopic robots. They have genetic blueprints inside them, and they respond to bioelectric signals, like how our nerves use electricity to talk to each other.
Oh, wow. So we're making an electrical circuit, but instead of wires, it's cells and bioelectricity.
Yeah, exactly. It's building with biological Legos. The AI is like the master builder, and the circuit cells are like the bricks.
Okay, I'm with you so far. But where do these circuit cells actually do their thing? How do they go from just individual cells to a whole creature?
Okay, well, that's where the toesis comes in. It's this special environment, a controlled space where we deploy the circuit cells in stages.
So the toesis is like the construction site for the organism.
Yeah, that's a good way to put it. Think of it as a high-tech womb or a bioconstruction zone.
Okay, that makes sense. But how do we make sure the cells stay on track? How do we get them to build the organism the right way?
Right. Well, that's where we get into etheric rhythms. It's basically a self-driving system for the organism's growth.
Oh, wow. The self-driving system.
Yeah, it keeps making adjustments to keep the development process going in the right direction.
So it's like a constant feedback loop, making sure the organism stays true to the original drawing.
Exactly. The rhythms are basically algorithms that control the bioelectric signals going to the circuit cells. They make sure the cells are dividing and growing and arranging themselves in the right way at the right time.
Wow, that's incredible. But wouldn't you need tons of different types of circuit cells to create all the different tissues and organs in an organism?
Yeah.
Like, how do you even manage that level of complexity?
Well, that's where it gets really interesting. This system uses pre-trained surrogate cells.
Surrogate cells.
Yeah, so these cells are already programmed to develop into specific types of cells, like muscle cells or nerve cells. It's like having a toolbox full of specialized cells ready to go.
Oh, so you don't have to create custom cells for every little detail. You can just use these pre-trained cells.
Exactly. It's way more efficient, like using prefabricated parts to build a house.
Okay, so let me see if I got this straight. You draw the organism, the AI looks at the drawing and makes a blueprint. That blueprint guides how the pre-trained surrogate cells are put together inside the toesis, and the etheric rhythms keep everything on track.
Yeah, you got it. And the really cool thing is, with current tech, the success rate for creating simple structures is already pretty high. We're talking 80% or more for things like tissues or simple organs.
That's impressive. But what about something more complex, like our five-legged frog? What kind of success rate are we talking about there?
Well, that's where things get a little more tricky, but even then, the success rates are pretty promising.
So we could really create a five-legged frog today.
It's possible, yeah. It involves several steps. First, the AI takes the drawing and turns it into a biological blueprint. That step has almost a 99% success rate. AI is really good at that part.
Wow, 99%. So then what happens? What's the next step?
So then the circuit cells, using that blueprint, start to build the basic structure of the frog, including that extra leg. And the success rate for that is about 85% right now.
85%? That's still pretty good. So what's the hard part?
Well, the next challenge is making sure that fifth leg integrates properly with the rest of the frog's body.
Yeah.
We have to make sure things like the nerves, the muscles, the blood flow, all of that is working right.
Yeah, I can imagine that being pretty complicated. What's the success rate for that?
It is complicated, yeah. Right
Hmm.
now, it's about 70% success for that seamless integration of the fifth leg. But remember, this research is like the bleeding edge. Those rates will probably get way better in the future.
Wow, that's wild. So even with the challenges, we're talking about a pretty good chance of actually making a functional five-legged frog. I mean, just
Mm.
imagine what we could do if we refine this even more.
Oh, yeah, the potential is huge, huge. But okay, so let's step back for a second and go back to the process. We talked about the AI blueprint, the toasis, ethoric rhythms, but I'm curious about those pre-trained surrogate cells. How do those actually get placed in the developing organism?
Yeah, good question. It seems like it would have to be really precise, like the timing and placement. How does it manage that?
Well, it's all about bioelectric signals. I mean, cells aren't just passive building blocks, they actually communicate with each other through electrical and chemical signals. And the toasis, the environment in the toasis, lets us manipulate those signals to guide the cells where they need to go.
So it's like a biological GPS for the cells.
Yeah, exactly. That's
Yeah.
a good way to think about it. And those ethoric rhythms, remember those? They're a key part of this too. They're like the feedback loop, making sure the cells are responding right to the signals.
Wow, it's like this perfectly choreographed dance of cells, all guided by the AI and bioelectricity. It's amazing how we're learning to speak the language of biology and build these complex structures.
It is pretty amazing. It's almost like we're talking to the cells, telling them where to go, what to do.
Okay, so I think I'm getting the complexity here. But let's go back to those success rates again. You said simple structures like tissues are already being made with like 80% accuracy.
Yeah, at least 80%.
Yeah. And even for complex organisms, the success rates are pretty good. We talked about the five-legged frog, but you said limb regeneration in humans might even have better rates.
Potentially, yeah, because with limb regeneration, we're working with a biological framework that's already there.
Wait, hold on. Are you saying we could regrow a human limb with this? Like, right now?
It might be possible. Think about it. We already have the blueprint for our limbs in our DNA. The challenge is turning those regenerative pathways back on and then controlling the process.
Okay, so how would we even start to regrow a limb? Like, what would the steps be?
So let's say someone has lost an arm. The first thing we do is map the bioelectric pattern of their other arm. That would be like the guide, the reference point.
Yeah.
And we can do that with like 95% accuracy right now.
Okay, so we make a bioelectric map of the missing limb.
Got it.
But then how do you actually, make it grow back?
That's where those pre-trained surrogate cells come back in. We can use cells that are already programmed to become, bone cells, muscle cells, nerve cells, all the parts of the limb.
And the toasis would be where they grow and organize, right? Using the bioelectric map and ethoric rhythms.
You got it. The toasis is like a perfect incubator. It gives the cells everything they need to grow and develop.
But what about reactivating those regenerative pathways? Like you said, how do we do that?
Well, we're still figuring out exactly how regeneration works. But there are some promising techniques. For example, we can use synthetic RNA, basically introducing specific instructions into the cells to turn on those pathways.
Oh, wow. So it's like we're reminding the cells that they can regenerate.
Exactly. It's like a wake-up call. And we can use bioelectric stimulation to amplify those signals, too. That helps the cells grow and differentiate more accurately.
This is incredible. We're talking about manipulating life itself to regrow limbs. That's amazing. What kind of success rates are we looking at for something like that?
It really varies for each stage, but the overall potential is pretty high. Like, reactivating the regenerative pathways could have 75–85% success, and then guiding the growth of the limb would be about 60–70%.
Wow, those are some really encouraging numbers.
Yeah,
I guess the hardest part would be making sure the new limb actually works with the rest of the body, right? Seamless integration.
exactly, yeah.
Yeah.
Integration's a big one. That involves nerve connections, muscle coordination, sensory feedback, all of that. And right now we're at about 50–60% success for that stage.
50–60%, that's still pretty amazing considering how complex this is. It seems like the toughest part is that fine-tuning, making sure the regrown limb is fully functional.
Yeah, you're right. And that's what a lot of the research is focused on now, refining our understanding of bioelectricity, improving the accuracy of the circuit cell tech, developing better AI algorithms to guide the whole process.
It's clear this technology could completely change medicine, but it also raises some ethical questions. I mean, if we can regrow limbs, what's to stop us from going beyond natural limits? Yeah, enhancing humans.
Yeah, that's a valid concern. The same tech that can heal can also be used for enhancement, and that brings up a whole new set of ethical problems.
Right, so it's awesome that this tech has so much potential for healing, But we have to develop it responsibly and really think about how it could impact society. This isn't just a conversation for scientists. It's for ethicists, policymakers, the public, everyone.
Absolutely. We have to talk openly and honestly about the benefits and the risks. We need a lot of different perspectives.
So this deep dive has definitely given me a lot to think about. It's mind-blowing that we might be able to draw life into existence.
It really is. And it just shows how important it is to stay informed about all this new science and tech. Keep asking questions, stay curious.
I mean, it's crazy to think we might actually be able to draw a five-legged frog and then make it real in a lab. It's like, where does imagination end and reality begin?
I know, right? It's wild. And as we keep pushing the limits of bioengineering, we're only going to see more and more mind-bending stuff like this.
Yeah, for sure. We talked about some of the ethical stuff already, but I think it's worth digging a little deeper. What are some of the potential downsides, the risks of this technology?
Well, one of the big ones is unintended consequences. These are really complex systems we're messing with. We can't always predict how these engineered organisms will interact with the environment or even like how they'll evolve over time.
So, like, we could accidentally create a new invasive species,
Yeah.
mess up a whole ecosystem.
Yeah, that's a real possibility. We have to be really careful. That's why testing and containment are so important. And then there's the whole ethical question of just manipulating life itself.
What do you mean by that?
Well, some people might say creating new life forms, changing existing ones, it's like playing God. Is it really our right to be messing with the building blocks of life like that?
Yeah, those are some heavy questions. And it's not just about creating new life. What about misuse of this tech? Could it be used for, like, bioweapons or designer babies?
Yeah, unfortunately, any powerful technology can be used for bad stuff. That's why we need strong ethical guidelines, regulations, make sure it's not being misused.
So it's not enough to just focus on the science, right? We have to think about how we use it as a society, what kind of safeguards we put in place.
Exactly. We need to be talking about this stuff, the benefits, the risks, and we need to hear from everyone, not just scientists.
It feels like we're at the start of a whole new era of science, right?
Yeah.
One with incredible potential, but also a lot of responsibility.
Definitely. It's exciting and a little bit scary at the same time. We need wisdom and compassion to guide us as we move forward with this stuff.
This deep dive has really been eye-opening. I'm still wrapping my head around everything we talked about. The fact that we might actually be able to draw life into existence is just incredible.
I know, it's amazing. And it just shows how important it is to stay curious, Keep learning and asking questions about this crazy world we live in.
Well, thanks for joining us on this deep dive into the world of bioengineering. It's a wild ride. Until next time, keep exploring, keep questioning, keep wondering.